Okay, hi everyone. We were working our way in from the outside of the cell toward the inside of the cell through all of these cell structures, and we've come to the membrane. Um, the plasma membrane, so we're talking about the plasma membrane of the cell, is composed of phospholipids, which you see here in this figure, and then also proteins. Okay, and the proteins can be associated with the membrane peripherally on the inside or the outside of the membrane, or they can be transmembrane spanning. Okay, and we'll see an image of that in just a minute. So the composition of the membrane would be phospholipids and proteins. The membrane has a fluid mosaic model. Okay, I mentioned that before, where the consistency of the membrane is about the consistency of olive oil. So it has a fluid consistency. And the proteins, okay, especially if they're not anchored to a cytoskeleton, they can move translationally within the membrane, okay, but they might be anchored to something. And the phospholipids can move translationally within the membrane as well. Okay, the phospholipids make a membrane bilayer, and they can kind of bob around. Okay, like if you think of like a buoy in the sea, it can bob around and move around a little bit, but they hardly ever flip through the membrane, because they have this like polarity almost, where one part of the phospholipid is hydrophilic, okay, which means water loving, and so it's going to interact with the outside of the cell as well as with the cytoplasm within the cell. And then you have the hydrophobic tails, okay, which are going to want to be, they're water hating, so they're going to want to be hidden away from the water, so they would be on the inside of that membrane bilayer. They would be kind of buried on the inside of that membrane bilayer. So if you had pure phospholipids and you mixed them with water, okay, they would make these micelles, okay, which are like a lot like cells, in that they are these spheres. There's water on the inside, there's water on the outside, and the hydrophobic part of the phospholipids would be on the inside of that membrane. So they would configure themselves so that they wouldn't be in contact with the water. Okay, so a matter of form, fitting, function, okay, which definitely is a huge theme in biology. The function of the membrane is to allow some things to pass, okay, and then to keep other things out, okay? So that would be selective permeability. Some things are going to pass through the membrane more easily than others, okay? And those other things, all right, that don't easily move through the membrane, you might need to have some type of transport protein, okay, or some type of carrier protein to be able to move that substance into the cell. All right. We mentioned with the outer membrane, okay, not to be confused with the cell membrane, but the outer membrane of the gram-negative. So the gram-negative cell would have a cell membrane, a thin layer of peptidoglycan, and then an outer membrane. That outer membrane was selectively permeable too. So this selective permeability is really a feature of all membranes. And with the outer membrane, you might remember me saying that it prevented antibiotics and enzymes, okay, like lysozyme that we talked a little about, and also penicillin, from entering into the cell. Okay, so gram-negative bacteria are actually resistant to penicillin. So here's the membrane bilayer, and you can see, okay, the blue hydrophilic heads of the phospholipids, and then you see those two little, they look like spaghetti noodles hanging down. Those are the hydrophobic tails, right? So all of the hydrophobic tails, okay, are buried in this membrane bilayer, and what's exposed on the inside of the sphere and on the outside of the cell, basically, would be the hydrophilic, water-loving head of the phospholipid, which could interact with water. So the fatty acid tail, okay, which is mostly just hydrocarbon, right? That is not going to mix well with water. Okay, so that needs to be buried on the inside of the membrane. You can see those two different types of proteins here. There are peripheral proteins, okay, which can be associated on the outside of the membrane, okay, or the inside of the membrane. They can be associated with the membrane itself, or with a transmembrane spanning protein. Okay, so the peripheral protein that you see on the outside of the cell is just associated with the membrane, whereas the peripheral protein that you see on the inside of the cell is actually associated with the transmembrane spanning protein. So there are transmembrane spanning proteins. There is a channel protein, okay, like that we were talking about, that could be used for some type of transport. You can see that there are glycoproteins, right? The proteins could have carbohydrate associated with them. Okay, also there are glycolipids, okay, so the lipids could have carbohydrate associated with them. So all of these different molecules make up the plasma membrane, okay, which has a fluid mosaic, a fluid consistency, and the mosaic part refers to the proteins that make up, so every different species, or, like in eukaryotes, every different cell, okay, that's specialized for a different function would express a different mosaic of proteins on its surface. Okay, I just want to review passive and active transport, and we'll go through some examples. But the membrane is selectively permeable, okay, which means that some things pass through the membrane more easily than others. So if we were in person, I would ask you what kind of things pass through the membrane easily. Okay, and someone would probably say oxygen. Okay, yes, that's true. Oxygen can pass through the membrane easily. Also, carbon dioxide can pass through the membrane easily. And the reason why is because those molecules are small and they're non-polar. Okay? Someone might even answer that question that water could move through the membrane easily. Okay, but that's not exactly true. Water is polar, okay? If you remember that the polarity, right? That water has a partial positive charges on its hydrogen atoms and a partial negative charge on its oxygen atom. Okay, and so that polarity actually prevents it from going through the hydrophobic core of the membrane. Okay, so water would go from where it's at high concentration to where it's at low concentration. Okay, but it would require some type of membrane protein that has kind of a polar center to it so that it could move through that polar center, okay, because it wouldn't be able to move through the hydrophobic core of the membrane. All right, so it would still be considered diffusion, moving from high to low concentration, but it would be considered facilitated diffusion because you require some type of transport protein. Active transport is different, okay? Active transport is not following the concentration gradient. Active transport is creating a concentration gradient. So you are moving things up their concentration gradient and kind of concentrating them on one side of the membrane. Okay, and we're going to see that for active transport, not only do you require some type of channel carrier type protein within the membrane, but you are also going to use energy. Okay, so sometimes we say that that is requiring work, okay, that you would use energy. Okay, so let's start by talking about diffusion. So diffusion is the tendency for particles to spread out evenly in the space available to them. So if we were to take this flask that was full of some gas molecules and put it into a larger container, it would move, those gas molecules would move from where they're at high concentration to where they're at low concentration. Okay, so here, and they would keep moving all around. Okay, some of them might even move back into the flask, and then they'd move out into the jar, and eventually they would be evenly spread out. Okay, and that would be called like reaching a state of equilibrium. Okay, they're still moving all around, but they're spread out. And basically here, the concentration gradient, okay, these molecules moving down their concentration gradient is the driving force. Okay, so no energy is required. Okay, so no work, no energy in the form of ATP is required. Okay, so for facilitated diffusion, it's still diffusion, okay? So the concentration gradient is still the driving force. No work is required. No energy in the form of ATP is required. But, okay, this would be like for water where the substance is polar, okay, partially polar. And so it's not going to move through the hydrophobic core of the membrane. So you require a pored transmembrane spanning protein. And then that transporter will allow the substance, okay, in the case of water, because of its polarity, okay, or maybe the substance is big, all right, or it has a charge, okay, it could be an ion, all right, so it's not going to diffuse through the membrane, okay, the ion would be charged, okay, that wouldn't go well with the hydrophobic core of the membrane, okay, something that's big is not going to easily diffuse through the membrane, but it could go through this transporter, okay? So you're still going to go from where you're at high concentration to where you're at low concentration, okay? But you're going to do so through a transporter protein, okay? This is what's called facilitated diffusion. Osmosis is the diffusion of water, and it usually involves a selectively permeable membrane. So the membrane would be permeable to water, okay, but not to the solute. All right, so here the solute would be that 1% sugar, okay, and then the sugar mixed with the water makes the solution, okay, so the water would be the solvent. All right. Okay, so in this example, if you have a 1% sugar solution on one side of a selectively permeable membrane, and you have no sugar, okay, distilled water, so that would be 100% water on the other side of the selectively permeable membrane, the water is going to move down its concentration gradient. So the distilled water would be like 100% water, whereas the 1% sugar solution, 1% would be the sugar, 99% would be water. So the movement of water would be toward the 1% sugar solution. And that water will move through that selectively permeable membrane until all of it moves through the selectively permeable membrane. Now, there is a way to prevent the movement of pure water into a solution containing some solutes, and that would be by osmotic pressure. So if something prevents, okay, if there's some type of pressure that prevents the movement of pure water into some type of solution, that would be exerting osmotic pressure. We talked a little bit about the cell wall preventing osmotic lysis. So bacteria are always in a situation where that's kind of like this, okay, where their solute concentration is higher than the surrounding environment, okay, so they're like the 1% sugar solution, and the environment that they're in is like the distilled water. So water is always moving into the cell, and the cell is, expands. Now, if it just continues to take on water, it's going to expand and rupture, right? Okay, but that's where the cell wall, okay, the peptidoglycan, okay, for the gram-positive, the peptidoglycan, and the outer membrane for the gram-negative, come into play, okay, in preventing that type of lysis, okay, or rupture. So the water moves into the cell and the cell expands, but then it stops expanding. And because the membrane is supported by the cell wall, okay, so it expands until it hits the cell wall, and then it stops expanding, okay, and hypotonic lysis is prevented. Okay, so there's always some osmotic pressure within the cell to prevent hypotonic lysis. Okay, just reviewing those tonicities. So what's normal for us would be for all of our cells to be in body fluids that are isotonic to our cells, okay, because our cells do not have a cell wall, okay. So if our cells were in a hypotonic solution and we're taking on water, our cells would rupture, okay. But like, for instance, our red blood cells in our plasma, the solute concentration within the cell and outside the cell in the plasma are the same. So water might move into the cell, but it would also move out of the cell at the same rate. So there would be no net uptake or loss of water. Now, that's what's normal for eukaryotic cells, okay, well, for animal cells, okay, but what's normal for plants, okay, and also for bacteria is to be in a hypotonic solution. So in a hypotonic solution, the solute concentration outside the cell is lower. Okay, so the water concentration would be higher. So the water would be moving into the cell, okay, down its concentration gradient. And what prevents hypotonic rupture or lysis would be the peptidoglycan cell wall. Okay, and then hypertonic solutions would be where the solute concentration outside of the cell is higher than inside of the cell. Okay, so the solute concentration is higher. Like, say for instance, if you put a cell into 10% salt, okay, water would move out of the cell and the cell would shrivel up. Okay, this is actually a special type of active transport, which is called group translocation. It's a special type of active transport for prokaryotes. Active transport in general is going to require energy and also some type of transport protein. In active transport, okay, you are moving a solute up its concentration gradient. Okay, so you're creating a concentration gradient. And remember, in passive transport, the concentration gradient was the energy, right? The potential energy that was used to reach equilibrium. And so you didn't need to put energy into diffusion, okay? But here, where we're creating, kind of an energetic situation by making this concentration gradient, okay, which has potential energy, we're going to need to use energy, okay? And the cell's form of energy would be ATP, okay? So you can see, all right, as the cell is taking up the nutrient, okay, and maybe it's like glucose or something, the cell's favorite carbon and energy source. So it's taking up the nutrient and concentrating it. Okay, you can see that there's more of the nutrient on the inside of the cell than on the outside of the cell. So it's taking up the nutrient and concentrating it on the inside of the cell and using ATP, okay, while it does that, all right? So group translocation is a special form of active transport that only occurs in prokaryotes. And what happens is that as the substance is brought into the cell, it is chemically altered so that it won't leave the cell. Okay, so like in this situation, okay, if this were glucose, the glucose could be phosphorylated, and then it would be less likely to move back out of the cell. Okay, so that's referred to as group translocation. Okay, so we're moving from, the outside of the cell toward the inside of the cell, and now we're actually inside of the plasma membrane of the cell. And I wanted to talk about some internal cell structures. So we'll talk about several internal cell structures, including the ribosomes, okay, the nucleoid. We'll talk about some inclusions, Okay, and then also we'll talk about spores. All right, and we'll talk more about sporulation in lab as well. Okay, so ribosomes. We said that bacteria have a 70S ribosome, which is different from our ribosome. Our ribosome, eukaryotes have an 80S ribosome. Okay, and I mentioned that that 70S ribosome could be the target for antibiotics. Okay, and so it could shut down protein synthesis for the prokaryote, okay, which is infecting us, but not be a problem for us, right, the host. Okay, so that would be a really good target for an antibiotic. The S, I just want to mention, you don't need to worry too much about this, but the S stands for the Svedberg coefficient, and it's a sedimentation coefficient, so it's not a weight. And just to give you an example, okay, so it's a sedimentation coefficient is measuring the sedimentation of that protein during centrifugation. Okay, so you would put the protein into a gradient and spin it in a centrifuge, and then it would be given a sedimentation coefficient. So together, okay, because there are two parts or subunits to the ribosome. There's the small subunit and the large subunit. So together, those two subunits are 70S, okay? But apart, the large subunit is 50S, okay, sedimentation coefficient, and the small subunit is 30S. Okay, so you can see that that's not a weight. Okay, so so there are other factors that go into that sedimentation, and the reason why the 70S it's 70S for the two subunits together is that when those two subunits come together, they make they are more compact. Okay, and so they sediment a little bit faster than they do separately. Okay, the ribosome is made up of protein. Okay, and usually when you see it, it looks like, you know, a blob, how proteins are represented. But it also has within it ribosomal RNA, okay, or rRNA. And we'll see, okay, when we talk about translation, that rRNA, ribosomal RNA, actually plays a role in positioning the messenger RNA, okay, within correctly within the ribosome. Okay, so I also wanted to talk about the nucleoid. Okay, so you can see in this transmission electron micrograph, you might notice that TEM, transmission electron micrograph. Okay, so this is the electron micrograph where you're taking a cross section. Okay, and the shape for these cells would be bacillus. The arrangement would be diplobacilli. Okay, so they went through binary fission, but they remained attached. Okay, so this is a diplobacillus. The nucleoid is where it contains the bacterial chromosome. So the bacterial chromosome is not just floating around within the cell, pinging off the walls. It is actually in a specific location within the cell, okay, but it's not membrane enclosed, okay, so it's not an organelle like it would be for a eukaryote. The chromosome is actually attached to the plasma membrane, and when we talk about binary fission, we'll talk about how the chromosome is replicated as the cell is, you know, increasing in size and making plasma membrane and peptidoglycan and becoming longer. And before you actually have the constriction that creates the two daughter cells, the cell is elongated, and with the two copies, okay, because the chromosome is being replicated, those two copies are kind of being teased apart, and each copy of the chromosome is attached to the plasma membrane, the plasma membrane, and as the cell is elongated, that is actually facilitating the segregation of those two daughter chromosomes to the two daughter cells. So it's almost as though the plasma membrane here is playing a role in binary fission to segregate the chromosomes like the mitotic spindle does in mitosis. Okay, I also wanted to talk about some cytoplasmic inclusions. So this is a really interesting cell. You can see the photosynthetic membranes. Okay, remember we talked about those, the chromatophores. So that would be where you would have all of the light capturing pigments and the photosystems, and so those internal photosynthetic membranes are providing surface area, okay, for all of those proteins and those molecules that are necessary for photosynthesis. You can also see in this figure that there are some carbohydrate granules. Okay, so you can see those little round granules within the cell. Okay, those are inclusions. And granules, storage granules, are just granules that are created, in the case of the carbohydrate granule, by carbohydrates, okay, polysaccharides. There are also vesicles, and vesicles are membrane enclosed. Okay, but again, all right, they would have some type of nutrient inside of them. So for the carbohydrate granules, okay, that we can see in this figure right here, the cell is storing carbohydrates. Okay, and why would the cell want to store carbohydrates? Okay, well, carbohydrates can be used in respiration to make energy, right? So a polysaccharide could be hydrolyzed into individual glucose molecules, and glucose could be catabolized in cellular respiration to yield ATP. Okay, I also just want to plant this seed right now that in the case where you have enough energy, okay, that glucose could also be used as a carbon source. Okay, so we can also catabolize amino acids for energy. We can also catabolize lipids for energy, okay? And they are not going to start right at the beginning of glycolysis, but they're going to get fed into somewhere along the way. They're going to get fed into that pathway to make ATP. And say if we have enough ATP, but we need amino acids or we need lipids, we could actually siphon off some of those intermediates, okay, just for their structure, their carbon structures, and make them into amino acids or even carbohydrates or lipids. So, okay, not only would this carbohydrate be used for energy, but it could also be used for carbon. Um, so as far as the lipid, I mean, as far as the vesicles are concerned, um, there is a lipid inclusion that is a vesicle, which would be polybeta hydroxybutyric acid. So that would be the common lipid storage material in bacteria. And lipids can also be used for carbon and energy. Okay. Um, so the last inclusion that we'll talk about will be the endospore. Um, so there are genera, okay, that produce endospores. In the lab, we study two. Okay, both are gram-positive rods. One is the genus Clostridium. Okay, the other is the genus Bacillus. Okay, um, Clostridium. You guys might be familiar with the species Clostridium tetani, okay, which causes tetanus, or Clostridium botulinum, okay, which causes botulism, food poisoning, or Clostridium difficile, okay, which causes C. diff, a diarrheal disease that usually occurs after antibiotic treatment. Um, all of those species of the genus Clostridium are all anaerobic. Okay, they're all anaerobes. They prefer an environment without oxygen. Bacillus, the genus Bacillus, on the other hand, is an aerobe. Okay, and can also exist in some species can exist in the absence of oxygen. An example of a pathogen from the genus Bacillus would be Bacillus anthracis, okay, which causes anthrax. So endospores form when there is some type of nutrient that becomes lacking. So when you have some type of key nutrient that is limiting, the cell would be dividing, okay? And then all of a sudden, if say nitrogen is limiting, or oxygen is limiting, or the carbon source is limiting, the cell would look as though it was about to divide, but instead, okay, it would replicate its DNA, but instead of dividing, okay, that DNA would be surrounded by a membrane, and then the spore would develop. Okay, so there would be two membranes around that core, okay, which would contain the duplicated chromosome, okay, and it would also probably contain small amounts of RNA, ribosomes, enzymes, and then surrounding that would be a membrane, and then surrounding that membrane would be a second membrane. And in between those two membranes, okay, that would be what would be called the cortex. You would have a thick layer of peptidoglycan put down, okay? And then outside the cortex would be the spore coat. And the spore coat is actually made of a really strong keratin-like protein, okay, like keratin that makes up your fingernails. And so this whole structure is very resistant to harsh chemicals and disinfectants. And you may have heard of like anthrax spores being sent in the mail, or you may have heard of how like C. diff, if a patient has C. diff, they're in a special room, and and there are a bunch of precautions that are taken, right, and special cleaning protocols as well. Okay, that's because the spores are highly resistant to disinfectants. There was actually an island off of the coast of Wales, and during World War II, they tested anthrax as a biological weapon, and the island wasn't really an island where tourists went or where anybody lived. I think it was extremely windy on the island, but they tested anthrax there, and they were never able to put any living thing on the island, like sheep. They weren't able to put sheep on the island because they would immediately contract anthrax and die for years, okay, for like 50 years, until they finally made a really huge effort to go to that island and clean stuff up and remove a bunch of soil. And then at that point, the island became safe to inhabit again, okay, mostly by animals, right? But it just goes to show how highly resistant the endospore is. Okay, it's a very dehydrated structure, so there is no metabolism occurring. It's kind of like a resting state, okay, and the spore, once it's formed, can survive for an indefinite period of time. I think your book talks about spores that were germinated after 10,000 years, and and there are other unsubstantiated reports that even older spores have been germinated. So within the mother cell, okay, the spore would be considered an endospore. Okay, but once that mother cell dies, all right, you would have a free spore. Okay, and when that free spore germinates and returns to the vegetative state, okay, of the cell growing and dividing, that is referred to as germination. It's not reproduction, okay, because you started with one cell, it became a spore, and then the spore became one cell. So there was no increase in numbers. And so this is more a state of differentiation than it is reproduction. I just want to talk briefly about the endosymbiotic theory. The endosymbiotic theory is really interesting. And at the beginning of this lecture, we were talking about comparing and contrasting eukaryotes with prokaryotes. And we mentioned that in eukaryotes, there were extracellular DNAs in the mitochondria and also in chloroplasts. Okay, and that is really interesting. Why would there be DNA in the mitochondria and the chloroplast? Just to let you know, there is also ribosomes in the mitochondria and the chloroplasts. Now, we talked about viruses not being living things, right? Because they are acellular. Okay, they're not made of a cell. All right, they don't contain ribosomes. Okay, and they don't contain, in some cases, DNA as their genetic material. Okay, in the case of retroviruses, they have RNA as their genetic material. So the mitochondria and the chloroplast, they have a membrane, they have DNA, and they have ribosomes. Okay, so they basically have everything that could have made them their own living entities. Okay, and in fact, with the endosymbiotic theory, it's a very, very well supported theory. Okay, not a hypothesis. There's like a large body of evidence to support this theory, that the mitochondria and the chloroplasts were once their own living things. Okay. And then they had an endosymbiotic relationship with a larger prokaryote, and where they lived inside of that larger prokaryote and eventually became the organelles. And that endosymbiotic relationship between two prokaryotes evolved into a eukaryotic organism. So the endosymbiotic theory is really describing how eukaryotes evolved. Okay. So life on Earth began about 4 billion years ago, and that life was prokaryotic, and the Earth was anaerobic. Okay, there was no oxygen on the Earth. The first eukaryotes evolved about 1.4 billion years ago. So the first life was prokaryotic, and then eukaryotes, unicellular, one-celled eukaryotes evolved about 1.4 billion years ago, so about 2.6 billion years later. So like I mentioned, that eukaryotic life evolved from an endosymbiotic relationship between two prokaryotic organisms. So there was one larger one and then one smaller one that lived inside. So symbiosis is the close association between organisms, and an endosymbiont would be the species that would be, excuse me, I sneezed, the species that would be living within the other species, which would be the host. Okay, so I just want to mention, the mitochondria. Let's just think about what the mitochondria does. Basically, for our purposes, we'll say that all cells have a mitochondria. Okay, not all eukaryotic cells have a chloroplast, right? That would only be the photosynthetic protists or plants that would be that would have chloroplasts. But all cells would have a mitochondria. Okay, so what does the mitochondria do? You make ATP, right? Okay, you make ATP with the mitochondria, and it uses oxygen, right? So oxygen would be reduced to water at the bottom of the electron transport chain. Okay, so oxygen is the final electron acceptor. Okay, so as the Earth is evolving and oxygen is becoming present in the atmosphere, a lot of anaerobic prokaryotic organisms actually died. And it was beneficial to have a symbiotic relationship with a prokaryotic organism that could use the toxic oxygen, in respiration, and also make ATP, right? That would definitely benefit the larger organism. And the smaller organism may have been benefited by the larger organism in that food was provided to it or some type of protection was provided to it. So that would be how that endosymbiotic relationship evolved. Eventually, that smaller prokaryote became the organelle of the larger prokaryote, which altogether evolved into the eukaryotic cell. This is just a partial list of evidence supporting the endosymbiotic theory. for example, okay, the size of the organelles, the mitochondria and the chloroplast are similar to prokaryotes. The mitochondria and the chloroplast, their DNA is not linear DNA, it's closed circular DNA like in prokaryotes. Also, like prokaryotes, the DNA lacks histones, which are DNA binding proteins that are found in eukaryotes. The ribosome that is in the mitochondria and the chloroplasts is a 70S ribosome, not an 80S eukaryotic ribosome. And the organelles have two membranes, okay, which is consistent with a smaller organism being engulfed by a larger organism and being brought into a vessel. A vesicle. Okay, so the vesicle would be one membrane, and then the second membrane would be the membrane of the cell. All right, and it turns out when they analyze these membranes that the inner membrane lacks the sterols, okay, like we mentioned, that prokaryotes do not have cholesterol in their membrane. All right, and finally, the organelles divide by a process that is a lot like binary fission. Okay, so there's a lot of really good data to support the idea of endosymbiosis of two prokaryotes evolving into a eukaryotic organism. All right, so this is the end of chapter 4, part 2, the narration.